Good morning, students. It was Kaushik. Kaushik, was it you who wanted to discuss if the Viva's marks? I asked you to inquire, right? From your batchmates. Can you please unmute yourself, Kaushik?
Yes, ma'am.
What is the decision regarding the increase in weightage of Viva?
Yes, ma'am. Uh, all all the student decided that 10 mark from Viva and 10 mark for each quiz.
What does that mean? I sorry, I couldn't get you. What is 10 marks for Viva and what is 10 marks for quiz? I didn't get that.
मैम पहले जो था मतलब वही फॉलो होगा ऐसा स्टूडेंट ने डिसाइड किया सभी ने.
नहीं करना है, Viva बढ़ाना नहीं है.
नहीं मैम.
अच्छा, ठीक है. तो क्विज तो एनीवे 10 मार्क्स का था ना?
हाँ मैम.
है ना? क्विज 1 10 मार्क का था, क्विज 2 10 मार्क का था एंड वाइवा वाज फॉर 10 मार्क्स, सो देयर विल बी नो चेंज, राइट?
यस मैम.
Okay, good. Okay, then I will see. So what I notice is that the number of students attending the synchronous mode is going down drastically, which is not an issue for me, but I guess it would always help students to join in the synchronous mode, especially because I can clarify a lot of doubt right at the moment when I'm teaching the course. And I will not have any other help sessions because if you can sign in, you can also to ask me any doubt. And this is I'm actually putting it on record here. Okay, so we will start with, I could not complete the topic in the last class for the simple reason that I ended the PPT 15 minutes before time to just have an interaction with you and to see what is it that's not clear, as I was teaching some things about nucleic acid and I knew that it might be a little too much for you if you don't ask me questions. You all are still not in the mode of raising hands and asking me questions when you have a doubt, but I strongly encourage you start doing it. It's high time you start doing it actually. Let's start with the small assignments that I gave you. Can anybody tell me what is the average GC content of human chromosomes? हाँ कौशिक.
Ma'am, the average GC content in human genome range from 35% to 60% across 100 kb fragment with a mean of 41%.
Okay, Ashi, is that correct?
Yes, ma'am. I saw the same answer.
So you know that I mean, it's as simple as clicking on the Google, right? But I just want you all to know, otherwise you wouldn't go and check it out. Anyway, the more important assignment was discussing how do you define a prokaryotic species. How many of you went back and or tried to find out what is a bacterial species or what is a prokaryotic species? I will wait for more hands, Kaushik and Ashi, and then I'll come back to you. Anybody else who, Vishantan, okay, hold on, just keen, fine. Any other hands? I see 13 participants. So four of you did look into what is meant by, so we'll start with Jaskeen.
Hello, am I audible?
Yes, you're audible. Tell me.
Ma'am, I searched for it and they said that bacteria does not classify criteria to be enabled in species because they keep on changing their genomes, and that's why they said that there could be a particular word like strain that can be used.
Is it Jaskeen? Is it that way? I mean, I would like to see where you read this, but anyway, Vishantan.
Mama has searched online and like I read a few papers, but I didn't get a satisfactory answer in any one of them. As Justin said, like most of the papers stated that like speciation of bacteria isn't as easy as for like eukaryotic cells. So and depending on the environment, the genomic content keeps changing. And like as you mentioned, that 70% similarity should be there at least. So, and like it's possible that for the similar kind of bacteria, more than 30 to 40% changes might occur depending on the environment in the genome. So, and
Okay.
because of this, new species will keep on being found because
Emerging.
of, yeah, rapidly, much more rapidly compared to eukaryotic cells.
Want to add something? Did you raise your hand by mistake or you had something to add?
It's almost the same thing. I mean, I served, but I didn't get a satisfactory answer. They said that the normal definition, that is interbreeding, doesn't apply. So, and some paper said something about, gram positive, gram negative strains, और मतलब ऐसे बहुत सारे criteria होते हैं. But मतलब कुछ I didn't get anything.
Okay, Kaushik,
Yes, ma'am.
you have raised your hand, so I thought you want to add something here.
Yes, ma'am. The definition of species in case of prokaryotic organism on the basis of determination of the DNA hybridization value and delta Tm value. And delta Tm is basically the difference between the melting temperature of DNA with drug and without the drug. And it is generally accepted that strain with approximately 70% or greater DNA relatedness with 5% or less delta Tm should be considered as member of the same species.
Hmm. Okay, so this is what we discussed in the last class, है ना? Vishantan, anything else that you would like to add?
Not exactly, ma'am, because I found one paper, but it was locked behind a subscription, so I couldn't read it. It
Okay.
looked promising, but I couldn't read it.
Okay, so you can lower your hand now. This is clear that the definition of a prokaryotic species does not abide by the definition of a eukaryotic species, for the simple reason that eukaryotic species are categorized on the basis of interbreeding, and that does not fulfill, prokaryotic species do not fulfill that criteria. So whatever definitions hold true are completely on the basis of the polyphasic taxonomy that we were discussing in the last class. Um, Parameters or cutoffs on the basis of nucleic acid hybridization and the basis of nucleic acid similarity is by far the best means of delineating a prokaryotic species. Okay, somebody initially stated that it's about, oh no, somebody wrote in the chat box last time that it's about DNA sequences. Yes, it is about DNA sequences. But I also mentioned that just having a similarity, let's say over 70%, does not mean that it's going to have, or a GC content falling within the number, does not mean that it's going to be the same species. If it is the same species, you will not find a deviation more than that. These are the cutoff values that we were talking of in the last class. So in the last class, out of the three components of taxonomy, that is classification, nomenclature, and identification, I tried to talk about what are the means by which organisms are classified, microorganisms specifically are classified, whether they are classical characters or they are molecular characters. Classical characters was easier in the sense that you had morphological, ecological, genetic, and physiological characters, and then we move to molecular characters by which you can classify microorganisms. Just you saw nucleic acid being one of the main marker by which you can identify a species or classify them into group, and the other marker was proteins. Proteins are usually more conserved compared to the nucleic acids because they have a functional role, majorly a functional role as well. So these were the two markers that we talked about, and we said polyphasic taxonomy is a taxonomy, as the name suggests, that would take into account all this phenetic characters, phylogenetic characters, as well as genotypic characters. So we talked about classification and we talked about identification. How do you go about saying that an organism is a novel species? It's not as straightforward as just going for a genome analysis. Then now we'll take up the third component, which is nomenclature. And you have seen these names being written in a particular way. Okay, scientific names you have to follow a certain rule to name, to write these scientific names. I'm just Okay, so like other organisms, bacterial names have two parts. They have a genus name and a species name. Both of them have to be italicized. And this type of a nomenclature, which is called as the binomial system of nomenclature, was given by Carl Linnaeus way back in 1753. In this case, the genus name is to start with a capital letter, and the species name is always written in small letters. And when you write the scientific names, you have to always underline. So when typing, it's italicized, but when you're writing it, it has to be underlined. And another thing that you would observe is after some genus names, maybe by now you would have observed in some of your reading material, that you will find either SP dot or SPP dot written after the genus name. Okay. I'll come back to that in a minute. Here I've just taken an example of E. coli. So Escherichia coli mein Escherichia is the generic name and coli is the species name. This is how you write it. Okay. And once you've referred to, let's say, the full name in the text, for example, you've mentioned that Escherichia coli does dash dash dash, the next time you can say, you can abbreviate the generic name with just the first letter. So it could be E. coli, E. coli. Now, about this, have you come across this two SP dot and SPP dot? Anybody in when you were reading through stuff or somewhere else, maybe reading some scientific articles? Have you seen this kind of a thing wherein the species name is not mentioned, but it's written as SP dot SPP dot? Anybody?
Yes, ma'am.
पता है इसका मतलब?
Not really.
Okay. तो ठीक है, नेक्स्ट टाइम पता करके आना. Next time आप मुझे बताओगे कि what is the difference between this missing P, okay, SP dot and SPP dot, what does it refer? There is a different meaning altogether once you write SP dot and SPP dot. ठीक है? Okay, so there is a Bible of microbiology called as Burgess Manual, which was published first in 1923, and it remains an authoritative reference for bacterial classification as well as taxonomy. So basically, there are two manuals. There's a Burgess Manual of Determinative Bacteriology, which helps in identifying bacterial species, and there's another one called as Burgess Manual of Systematic Bacteriology, wherein you have a comprehensive of all the as yet established taxa. And since 1980, the names of the new taxa are considered to be validly published only after they have been reviewed by the scientific community and published in this journal called as ITSEM, International Journal of Systematic and Evolutionary Microbiology. Only then you consider a name as accepted, a new taxa also as accepted. Okay, so after having described to you what are the consideration and the protocols to be followed for classification, identification, and naming of an organism, with emphasis on microorganism, I would now like to talk about the current classification system. After brushing upon some information that you might already have from your school times. So in the past, what started off as a two kingdom classification of living organism with just Plantae and Animalia, with as and when newer species were identified, especially microbial species, the two kingdom evolved to three kingdom classification. You see this group of Protista being introduced, and then it moved to the four kingdom classification, and eventually the widely accepted five kingdom classification, which you have probably covered during your school. Is that correct, students? Have you gone through the five kingdom classification?
Yes, ma'am.
Vishantan?
Yes, ma'am.
Yes.
Yes,
Okay.
ma'am.
Others? ठीक है. Very good. तो कोई मुझे बताएगा ये क्या था five kingdom classification? What were the five kingdoms?
Ma'am, plants, Plantae, Animalia, Protista, Monera, and fungi.
Monera, Protista, and fungi. Okay. ठीक हाँ. Okay. So this on who gave this classification?
Robert Whittaker.
Whittaker. Correct. Okay, so we will start with this first, Whitaker's five kingdom classification. You just mentioned to me all these five. What I wanted to tell you was that these five kingdom were classified on the basis of just these four features. Okay. So how these four features, now you know, cell structure matlab, प्रोकैरियोटिक या यूकैरियोटिक बॉडी ऑर्गेनाइजेशन मतलब यूनिसेलुलर या मल्टीसेलुलर मोड ऑफ न्यूट्रिशन मतलब whether they can make their own food or it's heterotrophic and reproduction meaning sexual or asexual. And when we've covered the other fungi, protista and bacteria, I try to emphasize on all these points. So if you would recollect, Monera, first they are prokaryotic, they are unicellular, they would have diverse nutrition, so they can actually photosynthesize as well as uh, okay, I thought somebody raised the hand. And here the reproduction is asexual. While in protista, okay, let's come back to protista later. Plantae, you will see that the cell structure is eukaryotic, is multicellular, they can make their own food and sexual reproduction is observed. Similarly, for Animalia, you will have eukaryotic, multicellular, but they cannot make their own food, they ingest food and the reproduction is sexual. Fungi mein tumne padha hai, it's eukaryotic, multicellular or unicellular. Mode of nutrition is saprophytic, that is they absorb, mostly they absorb their food, and the reproduction is sexual. I mentioned to you that Protista was kind of the junk drawer here, wherein there was a huge diversity in terms of, especially in terms of the body organization, in terms of the mode of nutrition, and also in terms of reproduction. So you will see absorptive, ingestive, photosynthetic organisms. You can have sexual as well as asexual reproduction. These are the 4 points that were taken into account when Whittaker gave this classification. Now, you will appreciate here that this was not polyphasic approach for classification with just these characters, these 4 characters being taken into account. There were a lot of ambiguities in the classification system. First, there was no distinct boundaries of Protista, Plantae, fungi. So you would see that the boundaries are quite blurred. You couldn't really define between the 3, especially Protista was too diverse. So anything and everything that did not fall under any of the other 4 would be put under Protista for without any scientific thought behind it. And the classification did not differentiate between archaea and bacteria. They were put under Monera. They were simply put under Monera. So after this 5 kingdom classification, there were further attempts to classify living organisms, and it so happened that it started diverging further. Okay, so Gray and Doolittle gave the 6 kingdom classification. Then there was a 7 kingdom classification followed by even an 8 kingdom classification. I'm told that the 8 kingdom classification is in the syllabus of, especially in some of the US universities, still being taught 8 kingdom classification. The most acceptable classification system was actually a modification of the 6 kingdom classification by Gray and Doolittle way back in 1982, wherein Carl Voess introduced the 3 domain into the classification, and this was largely based on looking at the molecular similarity of rRNA sequences. Now, I mentioned to you that rRNA can be used as an evolutionary chronometer or a clock. We've seen some of the differences even between bacteria, archaea, and eukarya. I mean, archaea we restricted to the cell membrane, but we really saw some of these differences. So, as of now, we accept that there are 3 domains: bacteria, eukarya, and archaea. And within these, you will find the different kingdoms. Okay, so before we end taxonomy, I would simply like to reinstate what I tried to explain earlier, and you got back to me with some of the information, and that is the endosymbiotic theory, which proposes that the organelles, mitochondria, and chloroplast in the eukaryotic cells were once free living bacteria. So at some point of time, they were ingested by another cell. So mitochondria is believed to have evolved from some free-living proteobacteria, while the plastids evolved from cyanobacteria. Yeah. There are various features of the two organelles which suggest this, and like I said, I just wanted to officially put into these slides. So some of the points in favor of the endosymbiotic theory include the fact that both mitochondria and chloroplast, they contain DNA, which is different from the DNA in the nucleus. In fact, it is similar to bacterial circular DNA that we saw in the prokaryotic cell structure. They are surrounded by their own plasma membrane. This we are aware of, similar to the bacterial membranes. And both these organelles, they reproduce or they divide through a process which is very similar to the asexual reproduction observed in bacteria. So you will find binary fission happening for division of mitochondria and chloroplast into newer organelles. And the internal structure and the biochemistry of chloroplast was similar to that of, it has been observed to be similar to that of cyanobacteria. While one more point that you saw was the porine-like structure present in the outer envelope of organelles, especially for mitochondria. So these are some of the points which indicate they actually are strong points in favor of the endosymbiotic theory. However, I told you to look into the points which actually refute the theory. I'm not sure how many of you went ahead and did that. अच्छा, इसमें एक point और होना चाहिए that the ribosomes that observe in the mitochondria and chloroplast are 70 mentioned here. Okay, so the pores and the 70s ribosomes are also points to be included. Which brings me to the end of this this topic. Here are some reads which make up interesting reads. You can go through these paper. Otherwise, I mentioned that Prescott is a good. It's a very easy reader-friendly book. Okay. Now, before I start the next topic, I would simply like to take you through just one thing. Give me a minute. How many of you have gone through the syllabus after I'd shown it to you? Did anybody go back and look at the syllabus? मैम कुछ एक्स्ट्रा तो नहीं पढ़ा रही हैं? किसी ने देखा है? तो एक बार हम लोग यहाँ देख लेते हैं and you tell me if things have been covered in the similar manners or the sequence in which it's written. We started off with our introduction to microbiology, the aims and scope, the history. I took a class on history and then also a class on role of the microbes in various sectors. Then we moved to the structure and function of the cell, wherein I covered first the prokaryotic cell and then the eukaryotic cell. And then we took up the distinguishing features of bacteria and of course archaea also. Fungi, algae. I said viruses I'm going to take up when we talk about bacterial genetics. Then in the last class and today's class, we covered up the third module, which is microbial taxonomy, wherein we talked about the classification of organisms and their nomenclature. And I introduced you to the two systems of classification. One is the Whittaker system and the other one is Boyce's system, which is currently being accepted. Currently, yeah, mostly accepted. Now I would like to move to the fourth module, which is about microbial growth and nutrition, wherein I'll start with nutrition. Okay. So because this is a relatively shorter topic, so we'll talk about nutrition first, and then from the next class on, we'll move to microbial growth. Is it fine, students? Do you have a question here with the sequence in which we're covering thing or anything related to the syllabus? Do you see where you're placed now? When I show you this. Bhavya Agarwal, can you confirm?
Yes.
Everything is as per the plan?
Yes.
Perfect. Acha, we are meeting in the evening also, है ना? I am the one taking your introduction class. कैसी presence होती है? तुम लोग सब synchronous attend करते हो या कोई नहीं होता है synchronous mode में?
मैम करते तो हैं पर पता नहीं।
करते हो पर पर मेरे में पता नहीं अच्छा
No, ma'am, it's
ठीक है
not like that, but
अच्छा
it's
okay
pretty
anyway
ambiguous
हाँ
every time. कभी कोई आ जाता है,
अच्छा
कभी कभी नहीं आता है.
I think 3 or 4 faculty members would have already presented if I'm not wrong.
Yeah, 3.
3 okay ठीक है fine. So I would now like to move to, if there are any questions from taxonomy, please ask me now, because then I'll move to the next topic, which is nutrition. Okay, everything is clear then. Let me share. तुम लोग को chat box में लिख रहे हो, एक मिनट. Justine ने लिखा है species and SPP dot just singular and plural. No, Justine, you have to really just Google it. हाँ बोलो देवप्रिया.
मैम टर्म पेपर का आपने बताया था डेडलाइन 14 नवंबर है, तो फिर उसका काम कब से शुरू करें हम लोग?
हम भेज देते हैं. तो
ओके मैम.
I would like to assign. I definitely cannot let you choose, right? Because तुम लोग choose करना चाहोगे. अगर हम लिस्ट दे दे तो ऐसा हो सकता है कि तुम लोग choose करो, या you want me to assign you the topics.
Ma'am, I guess it will be better if you assign, otherwise कोई
है ना?
तो
हाँ,
मजा
I
देने
got.
लगेगा.
कर दीजिए.
हम assign कर देंगे. मैंने 14th November दिया है, right? This is right after the
Minors.
minor one, है ना? So let me just see my calendar. Just give me a minute. Okay, major January में ना? January
ma'am.
Yes
1st.
मैम.
हाँ तो 14th मैंने दिया है, right? 14th मैंने तुम लोग को दिवाली के दिन दिया है. Are you sure?
मैम आपने तो यही दिया था 14
अच्छा
नवंबर.
ठीक है. 14 नवंबर मैंने दिया था, तो मुझे ऑब्वियसली हम इसको एक्सटेंड करने वाले हैं 21 नवंबर तक, क्योंकि दिवाली कैन नॉट बी अ डेडलाइन फॉर यू ऑल. ठीक है, दैट विल बी 21st, एंड इफ आई गिव यू द टॉपिक्स बाय दिस वीकेंड इट विल बी अप्रॉक्सिमेटली 4 weeks, राइट? नहीं, अब आज कौन सा दिन है? हाँ, या करेक्ट. सो यू विल हैव अप्रॉक्सिमेटली 4 weeks, right? ठीक है, I was working on it. It is just about the list of topics that I'm trying to finalize. ठीक है, fine. I'll list and also the instructions. Okay, so yeah, thanks for reminding. Okay then, let's start with the next topic. Ashi, bolo.
Ma'am, could you tell me what horizontal gene transfer is?
Okay, that is in fact that is one of the topics that we are going to cover in our bacterial genetics core module. But so in bacteria there is no sexual reproduction, right? But then there is variability. It is only when there is genetic variability that an organism evolves. Okay, one of the means of variability is to incorporate mutations. Mutations are random changes in the genetic matter. वो भी हम लोग बात करेंगे. You can understand that if an organism has been displaying so much of diversity and has evolved since so many millions of years, there has to be a means of generating genetic diversity, and sexual reproduction does not occur in bacteria, so there has to be some other means to generate such kind of a diversity. Now, some of the organisms can take up DNA from the outside and incorporate it into their own genome, thereby exhibiting genetic variation and evolving. Okay, so for example, I don't know how many of you know about it. What is very much in news right now is antibiotic resistant genes. So there are genes which are spread to different bacteria. It's not vertical. Transfer of gene vertically means being passed from the parent to the mother to the daughter. That is vertical transfer of gene. Horizontal transfer of genes is when the transfer is between a population or between different. So when I characters, which in taxonomy I said that it is possible to transfer genes between bacteria, but this is rarely cross genera. So within a genera, you will see genes being transferred between organisms. Okay, but this is not vertical as in mother to daughter, but this is across a population, in a population. आशी समझ में आया?
Yes, ma'am.
और इसके जो means हैं, there are three means of this kind of a genetic exchange, and we will take this up in the module bacterial genetics. ठीक है? We will go in depth with at least one of them. We will take it up in detail. Okay?
Okay, ma'am. Thank you, ma'am.
Okay. Can you see the PPT?
Yes, ma'am.
Okay, so like I said, this is a relatively short topic. I would just like to mention why it's important to know this first. Now, microbial nutrition, in order to be able to grow microorganisms in lab and to use them for any biotechnological application, we have to understand the nutrients that they require for growth, as well as what are the conditions in which they can be grown and how do they grow. So it's only after we have gained this knowledge that we can imitate the conditions in lab. Hence, this module is important for any biotechnology or biochemical engineering student. However, while I've mentioned this earlier, I have to stress again that till date, less than 10% of the microorganisms can be cultured, for the simple reason that we are still not aware of their nutritional requirements and other growth requirements in nature. How do they grow in nature? Okay. Now, the chemicals and the elements required for growth and for normal metabolism, they're called as nutrients. And if you look at the composition of a bacterial cell, you will see that 70% of it, and even a eukaryotic cell, mostly it is water. The biomolecules like proteins, like nucleic acids, lipids, and carbohydrates, they make up the remaining 30%, of which proteins form approximately half of the content. Now, when you talk about by a prokaryotic cell, there are certain elements called as major elements, which are required in a concentration of grams or mg per liter in the growth medium, which includes carbon, oxygen, hydrogen, nitrogen, and phosphorus, sulfur, of course. And you've come across all these elements in your biochemistry class. So C, of course, is a main constituent of cellular material. You know that oxygen is also a part of carbohydrates and other moieties. O2 is an electron acceptor in the anaerobic respiration. We will study this when we do the metabolic pathways. Nitrogen, you know, is present in amino acids. It's also a part of nucleic acids and various enzymes. Similarly, hydrogen. P kahan hota hai? Nucleic acids. You've seen some of the energy currency like ATP would also have P. So these are some of the major elements. The four biomolecules that you have studied or you are studying, either these elements have a role, a structural role to play or a functional role to play in these four biomolecules. However, there are other elements which are required by a cell in trace amounts or microgram concentrations. And these trace elements are required in such small amounts that they are present as contaminants in water or media components. So mostly you don't really need to add these trace elements in the media. As metal ions, the trace elements usually act as cofactors for essential enzymatic reactions in the cell. Many of them you will see are a part of the functionality of enzymes. Now tell me, how many of you have, from biochemistry, have you covered enzymes already? Proteins ho gaya hai?
Ma'am, proteins is done.
It's done. So enzymes as well. You know what cofactors, coenzymes are?
No, ma'am, not yet.
ठीक है, तो तुम लोग को पढ़ा दिया जाएगा. I guess that would be the next topic. They are important for the functionality of any enzyme. Certain metals are required for proper functioning of an enzyme. Also, what might be a requirement in trace amounts by one organism may be another's required element or a major element, okay, and vice versa. But usually the cations that qualify as trace elements in bacterial nutrition are like manganese, cobalt, zinc, copper, molybdenum, yeah, and some of them that I've listed. In some of the books, you will see that only the first 6 to still sulfur are considered as major elements. Then you consider them as minor element in certain books, but it's all about how it's categorized. And trace elements, of course, I said they are in micro amounts. Besides these major elements and the trace elements, which is also known as, like I said, you will probably see it as macronutrients and micronutrients. Okay, there are certain growth factors which are required for specific roles in biosynthesis, especially, and they are categorized into these 3 groups: purines and pyrimidines, amino acids, and vitamins. So while many bacteria can synthesize their own growth factors, some would require an external source of these growth factors. Like, for example, E. coli does not require any growth factor. It can synthesize all essential purines, pyrimidines, amino acids, vitamins, starting with their carbon source as part of their normal metabolism. While for others, it is needed. I mean, you need to externally apply it in the media. I've just listed down some of the functions of the growth factors, some vitamins and some hydroxamates. I've just listed this down for your benefit. Okay, so another question related to biochemistry. Have you covered the plasma membrane now? Lipids नहीं हुआ है. अच्छा, तो प्लाज्मा मेम्ब्रेन नहीं हुआ, इसका मतलब ट्रांसपोर्ट नहीं हुआ होगा. Fine. How does a prokaryote acquire these nutrients? So either the nutrients can be taken up passively, which does not require energy, or it could be taken up by an active mode, which involves utilization of energy. Now you're aware diffusion is the net movement of molecules along the concentration gradient. So any kind of diffusion here would bring in small molecules like N2, O2, water, and CO2. And these move across the plasma membrane by passive diffusion. So simple diffusion is what I'm talking about. In this case, there is no carrier protein involved, and once the concentration gradient disappears, as is very obvious, the net movement will stop. So across the bacterial membrane, if the concentration disappears, there will be no movement by simple diffusion method. And you can also understand that here there is no specificity, as there are no carrier proteins involved. So anything which is small and can cross this hydrophobic barrier will be transported through simple diffusion. In the case of facilitated diffusion, there are specific carrier proteins involved as permeases. Again, this is diffusion. It is facilitated, but it's still diffusion. So here also, if the concentration gradient disappears, the movement will cease as the movement is along the concentration gradient without the use of any energy. But here, because a carrier protein is involved, there is specificity in terms of which moiety is being transported across the plasma membrane. And for example, glycerol is one such moiety which is transferred through facilitated diffusion. So between the two, simple diffusion and facilitated, both do not require energy, but the facilitated diffusion is specific for a particular substance or a particular molecule. Now, under actual environmental condition, a cell does not always encounter a concentration gradient for transporting molecules. In fact, at times it has to indulge in transporting substances against the gradient as well. This is especially true for those molecules for which a cell would like to have a high concentration in the cytoplasm for its normal metabolism. So then it employs active transport, wherein movement takes place against the concentration gradient, but here it would then require metabolic energy, unlike in passive diffusion. Active transport is one of the most common methods used for the uptake of nutrients like sugars, like amino acids, organic acids, and many inorganic ions as well. And here, like facilitated diffusion, you will see that there are specific permeases or transporters, which are proteins spanning the entire membrane with specificity. And there are three main types of active transport: primary, secondary, and group translocation. We'll talk about each of the three now. So, primary active transport, also called as direct active transport or uniport, involves using energy. Of course, this is active transport, so there is a requirement of energy. And it pumps a solute across a membrane against its gradient. This is understood. Some of the examples are metal ions like Na plus, K plus, then some bivalent ions like magnesium, calcium 2 plus. These charged moieties, they require ion pumps or ion channels to cross the hydrophobic barrier. What you see here is a type of transporter called as ABC transporter or ATP binding cassette transporter with three different components. One is the membrane spanning protein that forms the physical pore across the cell membrane. It's also called as the carrier protein. The second element here is an ATP binding region that hydrolyzes ATP, providing the energy for this passage. And the third one is a substrate binding protein, which is a peripheral protein. अब membrane से तुम्हें याद होगा peripheral क्या होता है? Integral. So this is an integral protein. The carrier protein is integral, while the substrate binding protein is a peripheral protein that binds to the appropriate substance and moves it to the membrane spanning protein. Like you see here, it's actually attaching to that particular protein, bringing it to this to the substrate, bringing it to the carrier protein for transportation across the membrane. In gram-negative bacteria, the substrate binding protein is located in the cell's periplasm. याद है पेरीप्लाज्म क्या था? पेरीप्लाज्म ग्राम पॉजिटिव्स में होता है. हाँ बोलो, किसने अनम्यूट किया था? What is the periplasm? हाँ आशी?
The space between the two membrane, sorry, the peptidoglycan and the outer membrane in case of gram-negative bacteria, and periplasmic space is very thin in case of gram-positive bacteria.
I was in fact talking about one of the significance of the periplasm, wherein it scavenges these particular substrates to bring it into the cell, let's say Can you just confirm?
Ma'am, I went to the washroom for a bit.
Anybody else who heard me? Devpriya? Is that clear why it's called a secondary active transport process?
Ma'am, could you please explain it again?
Yes, ma'am. Please explain it again.
ठीक है. Primary active transport में क्या हो रहा था? The moiety which was being transported across the membrane through this carrier protein, the protein itself had a site for ATP binding. So energy was being utilized right at the point of the transfer. ठीक है, of the transporter which was mediating this transfer. ये clear था? This was called as primary active transport. Secondary active transport में there is an uncoupling between energy generated or energy utilized ट्रांसफर, द एक्चुअल ट्रांसफर प्रोसेस. और कैसे ये अनकपल्ड है हम आपको यहाँ समझा रहे थे. Through a normal process of the metabolic pathway, you will see, this is just one example, okay? I'm taking an example to show you what secondary active transport is. H plus का gradient, the cell is maintaining a gradient by a higher concentration outside by pumping. Whenever I say it is pumping, it means it is requiring energy. So H plus ka gradient has been created by the electron transport by pumping H plus outside. Forget about what process is involved. This has been created, generated. Haan, Satyakam?
Ma'am, I had a question, ma'am. In the primary active transport, there also must be something like electrical potential, like in the this secondary transport, right?
तो
मतलब उसमें
correct, बिल्कुल, you're right. Energy चाहिए होता है, whether it is the energy is derived from ATP or it is due to a potential gradient. Here, when you see H plus is being pumped out, this is uniport. ये समझ में आ रहा है? ये primary active transport ये uniport है ना? जो हम लोग यहाँ बात कर रहे थे, right? One moiety is being transferred with the expenditure of energy. That is the first part, H plus का expulsion. Okay. Now, when this has happened, it is because of this this gradient that has been formed. H plus can now even passively move inside. Because of this, along the gradient, it can move inside. But when it moves along the gradient through this particular transporter, the transporter is so designed that during its movement inside, Na plus can be thrown outside simultaneously. एंटीपोर्ट मैकेनिज्म से. इतना समझ में आया? अब समझ में आया आशी? ये यहाँ तक पहुंचे तो फिर हम आगे.
हाँ, यहाँ तक समझ आ गया.
तो यहाँ पे इस ट्रांसपोर्टर पे कोई एनर्जी कपलिंग हो नहीं रही है ना? डायरेक्टली इस ट्रांसपोर्टर पे देयर इज नो मींस ऑफ एनर्जी बीइंग यूटिलाइज्ड. व्हाट इज बीइंग यूटिलाइज्ड इज द ग्रेडियंट व्हिच हैज बीन जनरेटेड अर्लियर. करेक्ट? देवप्रिया, क्लियर?
Yes, ma'am.
Okay, so now that you see that this proton gradient which drives the sodium ion expulsion by this antiport mechanism is an example of secondary active transport. अब ये देखो, जब Na plus has been thrown outside, sodium binds to carrier protein complexes. Now these are all connected things, हाँ? H plus से कैसे सोडियम का एक्सपल्शन रिलेटेड है और कैसे सोडियम इज रिलेटेड टू द ट्रांसपोर्ट ऑफ शुगर इज व्हाट आई एम सेइंग. दिस इज अनदर एग्जांपल ऑफ सेकेंडरी एक्टिव ट्रांसपोर्ट. यहाँ पे एक हो चुका एंटीपोर्ट का एग्जांपल. नाउ आई एम सेइंग दैट Na plus binds to a carrier protein complex and it is upon binding of simultaneous binding of sugar. सेल को शुगर चाहिए अपने मेटाबॉलिज्म के लिए. ओके. The shape of the solute binding site changes and it binds to the solute, could be a sugar or an amino acid which is a requirement of the cell, and then by a symport mechanism the carrier's conformation alters so that sodium is released on the inside of the membrane together with the sugar or the amino acid which is brought inside. Antiport say sodium outside such that now this sodium ions can bind to a carrier protein which can simultaneously bring in sugar हो, चाहे वो अमीनो एसिड्स हो, चाहे कुछ भी हो. तो ये एक एग्जांपल एंटीपोर्ट का था और ये एक एग्जांपल सिम्पोर्ट का था. समझ में आया?
यस मैम.
ठीक है. और स्पेसिफिसिटी समझ में आई कि स्पेसिफिसिटी कैसे होती है एक्टिव ट्रांसपोर्ट में. ठीक है. Now maybe I will stop here. I will not take up the next or maybe I should. क्या करें? चलो group translocation बता देते हैं. तो primary active transport हो गया, secondary active transport हो गया. The third type, and then we will stop. This is different from primary and active transport in the sense that group translocation is a process in which a molecule is chemically modified as it is transported across the membrane. And one well-known example is that of a PTS system, which is phosphoenol pyruvate sugar phosphotransferase, which uses energy from a high energy molecule called phosphoenol pyruvate. Now, phosphoenol pyruvate, it serves as a donor. So when sugars are transported, they get phosphorylated by this phosphate group from phosphoenol pyruvate. And so this is what is happening. High energy PEP, it generates its P and glucose as it enters through this transporter. You have glucose 6-phosphate inside the cell. So here a complete group is translocated onto the moiety which is being transported. And in fact, we'll talk about this in this happens to be the first step in glycolysis. So when we take up bacterial metabolism, this is something that we will encounter. See that in group and was being transported. However, in group translocation, which is again an active transport, so there is a requirement of energy. There is specificity in what is being transported because of the presence of the carrier protein. However, when this sugar, glucose, this is just an example, when sugar is transported, it would enter the cell in a different form, in a modified form rather. Okay. So with that, you can ask me questions on whatever we talked about in microbial nutrition till now. And then in the next class, which happens to be next week on Tuesday, we will finish this topic and move on to microbial growth. Are there any questions, students? Okay, then we can stop the class. Thank you all very much, and we'll see each other today evening.